Hi, everyone. So, we've kind of talked about the basics of the endogenous immune system and how it's going to interact with cancer. In this video, we're going to talk about CAR T cells, which is a therapeutic technique that takes a patient's T cells. A patient's T cells are taken out of the body. They're genetically modified. And then they are injected back in and the result is these CAR T cells, which are these kind of new and improved versions of T cells that have kind of um, increased function and maybe an increased ability to fight cancer. So we're going to draw out kind of the three big modifications that happen with CAR T cells to start. Here's my CAR T cell. And again, all of these modifications are made genetically. So we alter the genetic code of this cell to change the expression of certain proteins. Maybe we add in the expression of certain molecules that wouldn't normally be expressed, or we upregulate or amplify the expression of molecules, or we could even downregulate the expression of molecules. So our first modification is going to be adding an antibody that is able to recognize kind of a specific antigen that's present on the surface of a cancer cell. So this is my cancer cell off in this corner here. So this antibody is specific to recognizing this antigen. And we've talked about how in prior videos how cancer cells can get rid of antigens that are being recognized. So we're going to make an antibody that can recognize this antigen. But one interesting thing here is that I've included this MHC1 molecule because normally we would need MHC1 to recognize this antigen. However, with these antibodies, that's not the case, so I can get rid of that. And we just need the presence of this antigen on the surface of this cancer cell in order to recognize it. So even if it's gotten rid of MHC1, we can still recognize this antigen. So that's kind of our first modification. Our second modification, if I draw it down here, is going to be a self-activating domain. So again, in prior videos, we talked about how T cells, or we've also called them CD8 plus cells, need to be activated by multiple molecules before they can attack the cancer cell. Here we're adding a self-activating domain that kind of skips over this step, and our T cell is able to activate itself. That's our second modification. And our third modification is going to be adding in more of these co-stimulatory domains. So normally we have these antigen presenting cells with these B7 molecules that stick up off the surface and interact with our co-stimulatory molecules. In this case, this is going to be CD28. Let me write that a bit bigger. CD28. And on our CAR T cells, we're not just going to express one or two of these. We're going to express many, many more than we normally would. So we've amplified this gene to express it a lot more frequently. Just say times 100, a lot more than we normally would. And we've added a bunch of these receptors to the cell surface so that we're increasing the chance of interacting with these B7 molecules on APC cells. That's our third modification that we're making. So again, to just write each of them down, we have adding an antibody that can recognize antigens without MHC1. We're going to add a self-activating domain. And lastly, we're going to add more co-stimulatory molecules. Right? So these are our three big adjustments that we're initially making to our CAR T cells. So these CAR T cells are more effective in attacking cancer cells, but cancer cells can still kind of fight back and attack these CAR T cells. So two modifications that cancer cells are going to make, I'm going to put them in, what color have I not used? I'm going to put them in blue to start. So the first modification that cancer cells can make in response to this is going to be getting rid of these antigens altogether. So we're going to get rid of these antigens completely. Let me maybe just circle and get rid of this. Getting rid of these antigens. So this is response 1. We get rid of antigens, right? And this is, again, our cancer cell doing this in response to these CAR T cells. Our second thing that cancer cells are going to be able to do, I'll stay up here so I can show this being done by our cancer cells, is they're going to cause something called T cell exhaustion. So cancer cells are going to start to secrete large amounts of this PD ligand. We've talked about PD ligand previously, where our T cells have a receptor for this called PD-1 receptor that can interact with this ligand. Normally, this ligand is present on the cell surface. In this case, we're going to be secreting soluble ligand, and it'll be binding to the PD-1 receptor and leading to this kind of inhibitory signaling inside the cell that's going to be downregulating the activity of these CAR T cells. That's going to be kind of response 2. Is decreasing CAR T activity with PD ligand. And cancer cells aren't going to be the only ones secreting this PD ligand. Cancer is going to get all of the surrounding cells, which we'll call the stroma. It's going to be secreting that PD-1 ligand as well to be able to interact with the PD receptor. And these are stroma that are being controlled by cancer cells, right? So we call these active stroma because they're being controlled by the cancer cells, right? So we talked about our 3 modifications to our CAR T cells. We talked about our 2 responses, response 1, response 2. where cancer cells are kind of fighting back. And our modification that we can make in response to this, kind of one final modification to talk about, is we can get rid of this PD-1 receptor, right? If cancer cells are targeting this PD-1 receptor, we can get rid of it. And that's kind of our final CAR T modification, is removing the PD-1 receptor to avoid this PD ligand T cell exhaustion. Right? So that's kind of our final step that we can take in this process. And there are, I guess you could say, fourth and fifth generation CAR T cells. There's been multiple generations of them. What's laid out on kind of so far is the first couple generations. For our fourth generations, we've kind of made some modifications. I can actually draw them down here to just separate it out entirely. So our fourth generation of CAR T cells is called TRUX. This is our fourth gen CAR T cells. And kind of the big modification that was made here is that they're going to secrete interleukin 12, which is an immunostimulatory molecule. And that's going to lead to increased immune activity and kind of decreased Treg activity. Which again, our Tregs are kind of those regulatory, somewhat inhibitory cells in our immune system. So that's our modification for TRUX for our fourth generation, is just adding that. For our fifth generation of CAR T cells, they don't have as fancy of a name. We're just going to call them fifth generation CAR T cells. The modification that we made is we tightly coupled activation with antigen binding. Right? And that's important because if our activation is not tied to antigen binding, which is that kind of like specific recognition step, then we can be activated and attacking cells that we're not supposed to attack, that don't have the antigens on them that would indicate that they're good targets. So this is to prevent kind of off target effects. All right. So that overall, if I zoom all the way out, is my overview of CAR T cells.